there aren't that many areas of paleontology, paleoanthropology that I think can really qualify as being a quote unquote paradigm shift in science. But I think this really qualifies as one because it basically changed everything.
Hello, I have the pleasure of speaking with Foundation Distinguished Professor Chris Beard of the University of Kansas. So welcome, Professor Beard. Could you please share a brief overview of your career and research?
Yeah, thanks for inviting me, Ian. It's nice to talk with you. So I started out my career, I was interested in fossils and paleoanthropology from a young age as an undergraduate at the University of North Carolina in Chapel Hill, North Carolina. I was lucky because just a few miles away was Duke University, which at that time had a lot of people that were studying fossils, including Elwyn Simons in the Fayum region of Egypt. Rich Kay was there, Matt Cartmel, so lots of kind of titans of the field. And I got kind of pulled into their orbit by kind of gravitational pull, I guess. So I got sucked into this business of primate paleontology as an undergraduate student. I went on to do my PhD at Johns Hopkins Medical School in Baltimore, where I studied with a couple of people with expertise in fossil primates, including Ken Rose, who was an expert on Eocene and even Paleocene primates and other mammals, and also Alan Walker, who was a close colleague of Richard Leakey's in East Africa. So after I finished my PhD degree, which was actually in anatomy at a medical school, so it was a little bit of a different path to where I am now than a lot of people follow. But once I finished my PhD at Johns Hopkins, I was lucky enough to get my first professional position. That was at the Carnegie Museum of Natural History in Pittsburgh, Pennsylvania. And I worked there for about 23 years. So it was a long time, and I really enjoyed my time there. It was the kind of position that gave me a lot of freedom and flexibility because I was not on a traditional academic calendar where I had to be in Pittsburgh during certain times of the year. There was a lot more flexibility in my position. That turned out to be critical because it allowed me to do field work in places, especially tropical countries and North Africa, during times of the year when if I had had a normal academic position, I probably could not have left campus at that time. So that was a critical time of my career. After 23 years on the curatorial staff of the Carnegie Museum, I moved to the University of Kansas, where I am now. I've been here since 2014, and have been able to continue the kinds of things that got me kind of started in the first place. So yeah, from an anatomy position in a med school to a curatorial position in a natural history museum. Now I'm an ecology, evolutionary biology professor here at KU. That's kind of the track that I've taken so far.
Thank you. What would you consider your most significant research achievement or the accomplishment that you're most proud of?
Well, in general, Ian, my idea has always been that in this field of paleontology and paleoanthropology, the most critical discoveries are always out there in the field, going out typically to a place and a time that is not well documented in the fossil record so far. So I've always tried to literally kind of fill in the gaps of the fossil record by trying to find things that no one has seen before. And that I think has generally proven to be a successful strategy. If I had to pick only one discovery out of all the years that I've been looking as being one that was maybe fundamental, I think it has to be Eosimias in China. At the time, that was it was a very timely discovery because it occurred at a time when the study of anthropoid origins was very much in flux. There were lots of debates about who anthropoids were related to on the evolutionary tree, when did anthropoids originate in terms of time, where did they originate in terms of geography, and basically what kind of transition from a non-anthropoid to an anthropoid, what did that biological transformation look like? And Eosimias, I think, answered many of those questions kind of all at once. So I think that that, to me, is probably the most important discovery of my career.
Is there a particular idea or perspective you hold that challenges widely accepted views in paleontology or paleoanthropology?
It's funny, looking back on it, I mean, when we first discovered Eosimias, it was incredibly controversial. I have to say that the first time that I made an actual scientific presentation at a conference about Eosimias, this was before it was ever published in Nature and then in Science, probably, I guess, maybe between 25 and 30% of the people in the audience seemed to like what I had to say, and pretty much everybody else thought that what I was saying didn't make a lot of sense. So it was very controversial when I first published it and first talked about it. But in the intervening years, I think a general and broad consensus has emerged that Eosimias is in fact a very basal anthropoid, and therefore it's not so controversial today. I think today, probably one area where my research and my findings are still, if anything, questioned by a large number of colleagues is my ideas about these so-called plesiadapiforms, which are, they're often viewed as very basal members of the primate radiation, things that are before the last common ancestor of living primates, but nevertheless on the line toward crown-clade primates. Ever since my PhD studies, I've basically believed that those things, in a purely cladistic sense, so in terms of recency of common ancestry, I've always thought that those things are actually more closely related to living flying lemurs of Southeast Asia, a different order, Dermoptera, than they are to primates. And there's a lot of skeletal evidence, mainly from the postcranial skeleton, but even some from the head, that suggests that that is true. And so I think that this is still very much controversial. It's probably about 50-50 now in the field in terms of whether people think that plesiadapiforms belong on the stem of Dermoptera or do they belong on the stem of primates. It's maybe not as important of a distinction as we once thought it was, because based on molecular genomics and based on our studies of paleontology and comparative anatomy, we now have a pretty good idea that the closest living relatives of primates are in fact flying lemurs, which was not the case when we started this whole thing about 30 years ago. It seems, I mean, there's still the question of precisely how do tree shrews fit into this whole thing, but I think in general the evidence in favor of a primate flying lemur clade, a clade that I would call Primatomorpha, is now supported by most people in the field. So that's a big advance, even if there are aspects of it that are still controversial.
You've already touched on this, but could you expand on how views on haploid evolution have shifted during your career, and how did you come to see Eosimias as pivotal to understanding anthropoid origins?
So let me just take a step back first, Ian, and let you know about how I started working in Asia in the first place. So I was a young curator at the Carnegie Museum of Natural History. I had an older colleague named Mary Dawson, who was an expert on fossil rodents and other mammals. And Mary had been working in various ways with Chinese scientists since the 80s, when China first started barely opening its door to outside collaboration. And so what happened was in the early 90s, a colleague of Mary's from the Institute of Vertebrate Paleontology and Paleoanthropology in Beijing, a man named Chi Tao, he contacted us and said, you know what, I found these incredibly interesting fissure filling sites in eastern China. We think they date to the Middle Eocene, about 45 million years ago. And we're finding lots and lots of new kinds of fossils from these fissure fillings. Some of them are primates. We're also finding rodents and lagomorphs and all kinds of different mammals. And he invited us to come over and have a look. And of course, I was interested. I had never worked in China before, but I knew that it had great opportunities and potential. And so I went there with the bias in my mind, Ian, that I would go to China, I would see Middle Eocene primates there, and they would be exactly like, or at least quite similar to the Middle Eocene primates that we knew about quite well from the Western United States and even from Western Europe. In other words, I thought that I would see a bunch of omomyids and adapiforms from the Middle Eocene. I never had any inkling that we would see something relevant to anthropoid origins. So we go there, we go to the site, we collect some fossils. There were more fossils in Beijing that had already been discovered. After going to the site, we went back to Beijing. We spent a couple of weeks working with our colleague Chi Tao to look carefully at these fossils. And I was trying my best to turn some of these fossils, which I immediately thought were very weird omomyids. I was trying my best to turn them into omomyids, and I was just coming into all kind of problems interpreting them as omomyids. And finally, I remember one morning I asked a colleague at the IVPP, I said, does anybody have a copy of Zelle and Delson 1979, the book called Evolutionary History of the Primates, which was like, at the time, it was the encyclopedia of every fossil primate that had ever been found, because I wanted to look at what early anthropoids looked like. I had never studied early anthropoids at that time, but I thought to myself, well, some of the weird aspects of these things that make them not look like omomyids, maybe they'll fit better as an anthropoid. So somebody says, yeah, I've got a copy. They run down the hall. They come back with their copy of Evolutionary History of the Primates. I open it up. We start looking at some of the earliest anthropoids at that time that had been described from places like Fayum in Egypt. And immediately I'm like, this is what these things are. I mean, they're primitive, but every anatomically derived character that these things have is unambiguous. They're always, the things that made them weird omomyids made them very, very primitive anthropoids. So I was convinced very rapidly that Eosimias being the first taxon was an early anthropoid. Later, that kind of started a number of joint field projects between the Carnegie Museum in the United States and the IVPP in Beijing. We went to multiple sites in China, including a site in central China in a place called the Yuanshu Basin in the valley of the Yellow River. And there we found incredibly complete material of Eosimias, which kind of sealed the deal that, yeah, this thing is a very, very primitive anthropoid. The received wisdom at the time, and one of the reasons that I went to China not looking for anthropoids, was that I was convinced that anthropoids had originated in Africa and that anthropoids had originated more recently, probably sometime near the Eocene-Oligocene boundary. All of these ideas, how did they come about? Partly because of the story of human evolution and ape evolution, which we know is rooted in Africa. And so people just kind of connected the dots and said, well, if humans and apes originated in Africa, anthropoids probably did too. And I think that the concept of a fairly recent origin for anthropoids was linked to old-fashioned scala naturae ideas about evolution. If you're primitive, you must have originated a long time ago. If you're more derived, like an anthropoid, you must have originated more recently. And so all of these strands were floating through all of our minds, I think, at that time. Eosimias upset the apple cart because it was not in Africa, it was in Asia, which at the time was unexpected. B, it was too old. It was Middle Eocene, probably 45 million years old. So about at least 10 million years older than the oldest anthropoids known at the time. and see its anatomy. I mean, just in terms of basic things like size, I mean, it was a teeny tiny primate, weighed about 100 grams if it were alive, but it also had some obviously primitive aspects of its anatomy. For example, instead of having a single lower mandible, it had the primitive mammalian condition of two dentaries that were not fused at the chin. Some aspects of its molar morphology, especially retaining an extra cusp on the lower molars called a paraconid, was not thought to be present in anthropoids. So there were lots of aspects of Eosimias that just did not agree with what we expected to find in an early anthropoid. But later through time, it started to make sense, especially, for example, when we started thinking in terms of anthropoid relationships and even primate relationships in terms of genomics. It's undisputed that tarsiers are the closest living relatives of anthropoids. We know that based on DNA sequences and various aspects of their biology, including having lost the tapetum lucidum reflective layer in the back of the eye. So where do tarsiers live today? Only offshore islands in Southeastern Asia. There's no record, there's no compelling record, shall we say, of tarsiers ever having lived in Africa. So if you're going to posit that anthropoids originated in Africa, the most immediate problem is, okay, well, what about tarsiers? Where did they originate? Because if we believe in the continuity of evolution, then tarsiers and anthropoids, back to the basic origin of both groups, they must have originated in the same place, right? It's hard to think that tarsiers originated in Asia, but anthropoids originated in Africa. That just doesn't make any sense. And so through time, I think it became more and more clear that Eosimias actually did agree with the big patterns that we saw emerging in mammalian genomics and in the primate fossil record, and that the anthropoid lineage is a very ancient one, even if for a long time we had very little fossil record of the early parts of that lineage.
And how long after these discoveries and descriptions in the literature did it take to convince other paleontologists and paleoanthropologists of the importance of Asia in anthropoid origins?
Yeah, I mean, honestly, Ian, it probably took 20 years for many colleagues to shift their ideas. And there are probably still a few people out there that still don't like the idea of Eosimias being a very early anthropoid. It is really, really primitive. There's no question about it. But I mean, I think what happened was early fossils in Africa, especially the earliest fossils from Fayum, which is really one of the best sequences of these early anthropoids in Africa. As older fossil anthropoids from Fayum began to be discovered and described, the anatomical gap between some of those fossils and Eosimias began to get narrower and narrower. So some of the animals, let's see, Proteopithecus is an example of an early fossil anthropoid from the Fayum. There's another one called Arsinoia. When you start to look at some of those fossils, you find that a lot of the features that Eosimias prominently has, paraconids on the lower molars, absence of fusion at the mandibular symphysis, small body size, a lot of these features also occur in things that everybody's happy to be an anthropoid, partly because they live in the Fayum, where lots of other anthropoids are found, and partly because they're slightly more derived than Eosimias is in terms of their anatomy. They look a little bit more like later African anthropoids than Eosimias does. So I think that made a big impact. I think the other thing that made a big impact on turning people around was the progressive discovery through time of additional fossils of Eosimias and its relatives in Asia. We started finding, for example, I think the discovery of ankle bones of Eosimias especially the astragalus and the calcaneus. That discovery, for people that weren't happy with the way that its teeth and jaws looked, that discovery changed some minds, because ankle bones of anthropoids actually don't look much like the ankle bones of things like omomyids and adapiforms. And Eosimias basically looked like a very primitive platyrrhine for all intents and purposes in terms of its ankle morphology. So I think that made a lot of sense. I think otherwise the discovery of slightly younger anthropoids in Asia, especially a fossil called Bahinia from Myanmar, I think that helped a lot of people believe that these Asian anthropoids probably were. There's this other big radiation from Southeast Asia, the amphipithecids, which are larger bodied animals and in some ways anatomically more derived. Those things seem to be another group of basal anthropoids from Asia. And so I think the combination of the Eosimiids on one hand and the amphipithecids on the other, as more fossils and more nearly complete fossils began to be discovered, more and more people started to see, oh, this looks like it could make sense. Plus, I mean, there literally was not, I mean, even for people that thought that anthropoids originated close to the Eocene-Oligocene boundary, the obvious question is, okay, from what? And there was a long history of people trying to link them up with adapiforms, which faced lots and lots of problems, because the more we studied adapiforms, the more tightly connected they seemed to be with living strepsirrhine primates, lemurs, and lorises. And that then became very difficult to reconcile with all of the biological evidence that linked anthropoids not with the strepsirrhines, but with tarsiers, the other group of haplorhines. So, and there was literally nothing in the African record before these very basal anthropoids from Fayum that could tell you what kind of other primates that you don't want to call an anthropoid, anthropoids must have evolved from. So I think as people started to come to grips with the idea that the anthropoid lineage itself could be very ancient, and with antiquity comes the possibility of much more primitive anatomy than what we considered previously, then Eosimias started to slot in there as making a lot of sense. It was the right age. Its anatomy was more or less correct for being a very basal anthropoid of that age. And once you realize that you've got to solve the tarsier anthropoid biogeographical problem, even its location in Asia started to make sense. So I think all of those factors together kind of made people reconsider anthropoid origins. So it's been a remarkable transformation. I think I mean, there aren't that many areas of paleontology, paleoanthropology that I think can really qualify as being a quote unquote paradigm shift in science. But I think this really qualifies as one because it basically changed everything.
You've already briefly mentioned omomyids and adapiforms, but could you summarize their evolutionary significance and potential relationships to living primates?
I mean, all of these areas are still active areas of study, so I don't want anybody that might watch this video to think, okay, well, we've got all that solved. I mean, there's lots of questions that are still out there to be addressed. In fact, in my lab, I've got a couple of PhD students that are working on some of these questions right now, so it's very timely. I mean, it's not easy, Ian, especially when we talk about omomyids, to answer the simple question, what do you think omomyids are? The problem is that omomyids, when people think of omomyids, I think we think of a large group and probably a disparate group of Eocene, mainly Eocene primates that tend to be smaller bodied. My strong sense is that omomyids, the concept is an oversimplification. So some things that are generally considered to be omomyids, I would nominate a taxon called Shoshonius. Some of those fossil primates, in my opinion, are clearly related to tarsiers. Tarsiers, I mean, let's just consider very quickly tarsiers, because they're probably the most special primates alive on the planet today. If a Martian biologist visited us and did a biotic survey of primates of the Earth, there are two taxa that the Martian would say, well, these are weird. And one would be Homo sapiens and the other one would be Tarsius. And they're weird for totally different reasons, but they're just out there. I mean, tarsiers have so much weird biology, so much weird anatomy about them that, you know, it ought to be an easy thing to say, okay, this fossil seems to be related to tarsiers, this fossil maybe not. But Shoshonius, which is about 50 million years old, from Wyoming, Shoshonius already shows hypertrophy of the orbital region like a tarsier. It's not quite as far gone as a tarsier is, but it's way beyond any other even nocturnal primate that we know about in terms of enlargement of the orbits. It has aspects of its the basicranial region that also show features that are only tarsier-like. So long story short, I think that at least some omomyids are clearly related to tarsiers. Other omomyids are really, really primitive. And I can't honestly say whether all omomyids are on the tarsier lineage or whether they maybe make a kind of a muddle down there close to the part of the tree where tarsiers and anthropoids finally come together. So I think it's a hodgepodge of things that we call omomyids. Some of them, no question, I think probably belong on that tarsier lineage. If we believe that that is true, then what that means is that the bifurcation between that big lineage that on the one hand is going to lead to tarsiers and the other lineage that on the other hand is going to lead to anthropoids has to be very, very ancient. If, for example, Shoshonius is truly on the tarsier side of that bifurcation, then anthropoids have to be around at least since 50 million years ago, and no one has ever seen a 50 million year old anthropoid. So we just don't know what those things would look like. So it opens all kinds of additional issues. So yeah, it's very, very complicated, but omomyids are a fertile field for primate paleontologists to study right now because so much new is being discovered about them.
And the adapiforms, you see them as potentially containing the common ancestor of living strepsirrhines, the lemurs and the lorises, or you see them as a more district-related sister grouping?
Actually suspect that the adapiform radiation, which was huge, I mean, my God, how many taxa of adapiforms have been described at this point? We're probably 50, 60 genera, something like that. They're known from Africa, from Asia, from Europe, from North America. So it's not surprising that this giant evolutionary radiation was as diverse, as disparate as the omomyid radiation was. But my strong sense is that within that big radiation of adapiforms are close relatives of lemurs and lorises. And it may be the case that some adapiforms are closer to lemurs and lorises than others are. So, you know, we call that kind of a group, a paraphyletic group as opposed to a monophyletic group, because they could constitute like a cone-like approximation of the crown clade that includes living lemurs and lorises. But there's no clear evidence that I know about that any adapiform would not belong on the strepsirrhine lineage some way, shape, or form, either very, very basal or perhaps a little bit higher up toward living lemurs and lorises. There's one group that one of my PhD students and I have been working on relatively recently, which is not a classical adapiform group. In fact, they were first described as omomyids, believe it or not. This is a fossil from North America called Egamuachashala, a terrible name, but the name was derived using the Lakota Sioux language because the first fossils were found on the Pine Ridge Reservation in South Dakota. For a long time, these things, they are actually known from the late Oligocene. So these are quite recent fossils. They're the most recent fossil primates from North America before Clovis man shows up in the archaeological record here. So Egamuachashala for a long time was thought to be an omomyid, very strange looking teeth, highly derived. But some recent discoveries in Asia, especially in Guangxi Zhuang Autonomous Region near the Chinese-Vietnamese border, clearly show that Egamuachashala is a fairly recent immigrant to North America from Asia. And with these Chinese fossils and with other fossils from places like Pakistan, it's pretty clear that Egamuachashala and its relatives are one group of adapiforms that just got to be very strange in Asia, and then they colonized North America after the Eocene-Oligocene boundary and the climatic crisis that occurred then.
Thank you. You've already touched on some of your fieldwork. I was wondering if you could go into detail of some of the logistics involved. And you've worked in a wide variety of places around the world. So how you go about identifying field sites, or kind of how the excavation methods have changed through your career and how they vary between different environments.
So Ian, let's circle back and let me remind everybody that my basic idea is that in order to make real advances in this field, we have to go fill in the fossil record in places and times that haven't been looked at. And, you know, especially in Europe and North America, because paleontology on those continents has been going on since the late 1800s, we found a lot. It's not to say that we can't find more in those continents, but the fossil record of Europe and North America is reasonably well established at this point. And so to make really pivotal new discoveries, we have to go increasingly to parts of the world that are off the beaten path, so to speak. And I've been really lucky. I don't know how, but somehow I've been able to have colleagues, especially international colleagues, that have facilitated my work in countries that aren't often visited, let's put it that way. So I guess the highest profile foreign field work that I've engaged in has been in Myanmar and also in Libya in North Africa. And both of those field projects were undertaken with my French collaborator, Jean-Jacques Jager. And I have to say, because of geopolitics, you know, the United States is an important geopolitical entity, but that comes with upside and downside. It often means that American geopolitical relationships are poor enough with certain countries that American scientists are it's almost impossible for us to work there. So literally, I was able to work in Myanmar and Libya thanks to my French colleagues. They established the projects, invited me to collaborate with them. And so I was able to work in Myanmar when it was run by the military junta. And I was able to work in Libya both before and after Muammar Gaddafi was running the country. And so you can imagine how complicated the logistics are to work in places like that. It's really not easy, and it can only be possible with a lot of work, not just by scientists, but in this case, actually by diplomats that are constantly working to try to make sure that the local government is okay with these foreign scientists coming and working there, and that we have all the proper permits and permissions and stuff to do what we want to do. So that I have to kind of lay that out first. But beyond that, I mean, I will say that even getting the permissions and permits and things to work in a place like Libya, that's a great first step. But after that, the organizational and logistical work that goes into mounting a full-scale expedition to the middle of the Sahara Desert is huge. When we worked at a place called Duratala, which is in south central Libya, just north of the border with Chad, we literally were about 300 miles from the closest tiny little settlement. I mean, there was nothing but sand in every direction there. So we had to bring everything. We had to bring all of our food. We had to bring all of our water. We had to bring big drums of gasoline for the vehicles. All of that had to be transported by large lorry trucks to our remote field camp near Duratala. And then we set up camp out there and spent every day searching for fossils along this big escarpment. The outcrop was quite nice. Everywhere where there wasn't a sand dune covering over the actual rock outcrops, you had access to the rocks and you had the potential to find fossils. The problem was that the rock outcrops were typically exposed on an almost vertical cliff face. And so you had to kind of, you would find a place that was producing fossils, and then you had to kind of dig a little trench so that you could hang on to the cliff face and then try to dig fossils out from one little layer. And of course, that worked for a few days, but the more you dug into that cliff face, the harder it was to get access to more of the fossiliferous sediment, because you're literally just going into the vertical cliff face. So eventually it becomes impossible. The other aspect of looking for fossils in the deep Sahara in Libya is, I told you we had to bring all of our water there. Well, the best way to find fossils of these early anthropoid primates, all of which weighed about 100 grams and were therefore tiny, was by this method called screen washing. To do screen washing, you've got to have water. And so we took probably half of the water that we transported out to the Central Sahara, and we made this kind of elaborate water recycling system where we could use the water to screen wash, which looks a lot like panning for gold, if you're familiar with that. But then normally when we do that under normal circumstances, the mud and stuff just goes away and we get fresh water to do more screen washing. Here we had to kind of let the mud dry, recycle the water, use it for a second round of screen washing. And it was very complicated, actually, to be able to screen wash in the middle of the Sahara Desert. But that's how we were able to find early fossil anthropoids in Libya that are still among the oldest fossil anthropoids known from Africa. Yeah, it's challenging. Honestly, if there's one place on Earth that I would be very happy to return to, because I think the scientific potential is so high, it would be Libya. But again, that's one of the countries that today it's still not stabilized after the Arab Spring uprisings. So it's kind of borderline a failed state at this point. So security is just not quite good enough to return to Libya and try to do this again. But I hope that it happens before my career is over, because I would love to go back.
So now is the quick fire round where I ask the same questions to all the guests in a particular subject area. So you've already effectively answered this earlier, but are anthropoids more closely related to tarsiers, omomyids, or adapiforms?
Well, it's a nuanced answer, because among living primates, no question, tarsiers are the closest relatives of anthropoids, but because some omomyids are specifically related to tarsiers, I guess I would say that it's a kind of a combination of omomyids / tarsier clade that's the closest relatives of anthropoids.
What do you think was the likely diet of the last common ancestor of all living primates?
Okay, so what we know about the fossil record is that the earliest primates were uniformly tiny, and by tiny I mean probably 100 grams maximum in terms of adult body mass. So when you're that little and you're a primate, your diet is fairly constrained. You've got to ingest lots of calories rather quickly. And so I think that these earliest primates would have been eating fruits, insects, and probably some sweet gum and exudates out of trees, kind of a combination of all of those items.
When do you estimate the last common ancestor of living primates lived?
It's an interesting question, Ian. I mean, based on what we currently know about the fossil record, I'm strongly inclined to say that the last common ancestor of living primates probably would have lived sometime during the Paleocene. So that's between 65 and, or sorry, about 66 and 56 million years ago. If we're just looking for a nice round number, how about 60? But that being said, it's possible that when we throw in the so-called stem lineage of primates, so fossils that are closer to primates than they are to the living sister group of primates, which would be flying lemurs, maybe you're getting really, really close to the Cretaceous-Paleocene boundary. One of the reasons that I'm very reluctant to go beyond that is there are credible studies that suggest that there are no bona fide placental mammals in the Cretaceous. I personally think that it's likely that there are placental mammals in the Cretaceous. Maybe we either haven't found them yet or we haven't adequately identified them. But until somebody really clearly shows that there's a late Cretaceous placental mammal, that means a crown-clade placental within the group of living placental mammals we know about today, it's kind of hard for me to imagine that primates go back that far. So yeah, probably sometime around 60 million years ago would be my guess. It's hard to imagine far back in the Mesozoic.
Thank you. And just a quick follow-up. You mentioned earlier about the likelihood of an early anthropoid ancestry, which would suggest a distant haplorine common ancestor. So does that mean you see a relatively short gap between the haplorine and steproline split and the origin of the haplorine lineage?
I think, Ian, that you've hit on a very important point. I think once the last common ancestor of all primates evolved, let's assume maybe 60 million years ago, there was not a long interval of common ancestry of those of strepsirrhines and haplorhines before the haplorhines are going to diverge. Perhaps even only a couple of million years. So I would be okay with last common ancestor of living primates being at, let's say, 60, and the last common ancestor of living haplorhines being 57, 58. There's not, I mean, it's one of the reasons I think that even in terms of genomics, if there's not a giant amount of evidence supporting haplorhiny, it almost comes out to be a trichotomy in terms of many analyses. I think haplorhiny is real and valid, but I think tarsiers and anthropoids have been separated a heck of a long time. Maybe that divergence is even still in the Paleocene. I wouldn't be surprised.
Thank you. And the final question is always something outside of your research directly. So I wanted to ask you about your thoughts on data accessibility in fossil primate research, for example, regarding scans, access to specimens, repositories, that type of thing. And what type of improvements would you like to see in the future?
Well, thankfully, there's a lot more accessibility today than there was even 10 years ago. Thankfully, because of digital morphology. I mean, with the invention of micro-CT scanning, we can now rapidly scan fossils. We can post detailed digital data on online repositories like Morphosource and others so that it's broadly available to the scientific community. I mean, when I was a graduate student, you know, some researchers would kind of hoard fossils and not let anybody look at them, and that obviously stymied, I think, scientific progress, because you know, we need to have a community of people looking at these specimens and trying to interpret them instead of having everything be very proprietary and stuff. So I think with the advent of digital morphology, we're rapidly getting away from that, so I mean I think that we're probably going to continue in that direction, and that's a very good thing for science and even for the public.
Thank you, Professor Beard, for doing this interview. It's been a pleasure speaking with you.
Really appreciate it, Ian. It looks like you've got a great project here. I'm going to have future generations of students look at your videos.